Welcome back to Biology B. This is lesson 1.3, Biotechnology and Engineering. If you remember, the big idea for this unit is that cells are specialized because even though they have the same DNA and genetic code, they don't always produce the same proteins, which causes them to have different functions. So in our learning targets or success criteria to reach today's lesson is to explain how DNA technology is used to construct recombinant DNA molecules to alter regular protein production, and to understand how genetic engineering, sometimes called biotechnology, is used to produce novel biomedical and agricultural products. So first we have to start by exploring, again, prokaryotic cells. When you think about prokaryotic cells, remember that common name for that is bacteria. Bacteria are single-celled organisms. They have no nucleus. They do have DNA, but they don't have a nucleus to hold it in. As opposed to you and I, which are multicellular eukaryotes, which have a nucleus. Other examples would be plants, animals, protists, and fungus. And they all have DNA, but they all have that nuclear envelope to contain it all in. When we're talking about bacteria or prokaryotes, what type of DNA do they have? They have what's called, they have circular DNA. And we're going to discuss what that's called later on in this lesson. They don't have long DNA. That would be eukaryotic. You have long DNA that you condense into chromosomes, if you remember that conversation. Sorry for my handwriting here. That's eukaryotes. Alrighty, plasmid is what we call bacterial DNA. It's a single circular piece, and it's in addition to the main chromosome. The plasmid, which is here, can self-replicate. And those are what we use when we are playing with another organism's DNA for recombinant DNA. When you compare prokaryotes versus eukaryotes, their DNA in a prokaryote has fewer genes just because there's physically less DNA. And again, it's a circular chromosome. And because there's less DNA, genes are found closer together. There's less non-transcribed regions. But it does still go through transcription at a ribosome, just like in the eukaryotes. The difference is the eukaryotes have more DNA, so more genes. The transcription requires helper proteins. We call them transcription factors. The genes are farther apart, and they have large sections of DNA that are not transcribed. Sometimes that's called junk DNA, and sometimes those have to be removed after the fact. But they both undergo the same process of protein synthesis, where the DNA is copied by RNA polymerase into an mRNA strand. The mRNA strands travel to a ribosome, and then the ribosome makes a copy of the eventual protein. When we talk about biotechnology, that's just life technology, right? So it's using living organisms to produce materials that we might not be able to make on our own. The example we're going to be talking about here in class is recombinant DNA. So if you break that word down, recombine DNA. So this is obviously not a real example here, but you've got a frog and a pear, a frere, I don't know, but that'll never happen in a real, obviously, situation. But we would use it in, say, vaccines, insulin, cancer therapies, or medicine for asthma treatments. Most of these things we don't make ourselves, or we don't make it efficiently. In order to in order to make this recombinant DNA, we use bacteria as the host for foreign DNA. Now, when we say foreign, that's usually ours, human, like one of our genes that we want to produce faster or more efficiently. And we do that because it's easy to put the gene into bacteria. They're very simple creatures. We just open them up and insert. We're going to go through the steps in a little bit. But it's easy to access their DNA because they're not in a nucleus, and they're easy to access everything inside because there's not many things inside. Bacteria grow very fast and they're very easy to cultivate. They reproduce very quickly. So every time this bacteria reproduces, just like the cell cycle we learned about, where the DNA is synthesized and then the cell divides. Now, remember, in mitosis, that's when bacteria go through as mitosis, but remember, at some point, the DNA is synthesized in the, I should say, the S phase, right? The S phase is where DNA replication happens. And then mitosis happens. And after mitosis, then you have two new cells. And each new cell will have the gene that you inserted. The reason, again, that we use bacteria is because they're tiny, they're easy, and they're simple. And they reproduce fast. How do we use recombinant DNA? Recombining DNA of different organisms. So actually, let's put the number here, because we're not recombining DNA from the same organism, it's from two different organisms. So if a bacterium is inserted with a human gene that codes for a human protein, the new gene will be added to the bacteria's genome. In other words, the bacteria doesn't recognize that there's a new piece of DNA. Bacteria doesn't have a brain. They don't think about things. They just reproduce their DNA. And if your DNA is stuck in their DNA, they don't recognize a difference. They're mindless. So when this plasmid is going to be replicated as this bacterial cell duplicates, it's going to duplicate this gene and produce in mass quantities the protein that we're looking for. So we use them for scientific research. We use them for medicine and pharmacology. We use them, yes, GMO. Here's the word, the acronym that's the big bad one, GMO, right? We use high temperature, we use it to make snow, which I didn't know, I just learned that. Oh, this one is really cool. We can make bacteria that can better process or clean up oil or toxic spills. We actually use that technology now. And then, of course, in the food industry, we make our food products bigger, better, stronger, healthier, live longer, make more meat, etc. Here are the steps to make recombined or recombinant DNA. This will be on the test as a sorting question, right? So you will have to know what order these steps are happening. So first things first, remove the gene to be donated. So that's this part here. You're taking from the DNA inside here before it's condensed from the donor organism. You take a little snip of the DNA that you're interested in, the isolated gene. That's step one. You have to remove the plasmid of the bacteria. Okay, that's step two, just removing it, isolating it, pulling it out so you can take it, so you can open it up later. So this is step two, opening it. That's step three. That's these two parts here. Insert the donor gene into the plasmid. So this is a recombinant DNA molecule. There's the word right there, right? This is a recombinant DNA molecule. It's a plasmid with a new DNA gene from another organism inserted. Now, once you've made the recombinant DNA, you can't just leave it hanging out there. It's not going to be reproduced out in the air. You've got to put it back into a new bacterium. You've killed this bacterium. You killed this bacterium in order to make this happen. So now you insert it into a new bacterium. And then every time, remember, every time this bacteria reproduces, it's going to make more of the same bacterium. It's going to have the same new plasmid in there with a new snip of DNA inside. Okay, so that's the important part. Remember, bacteria reproduce very, very fast. That's 5. So the last step then is the bacterium replicating, transcribing the protein or the DNA and translating it into a new protein. So make sure you know these steps in this order because you'll see these on a test. The biggest example that we use recombinant DNA for is insulin. So if you have ever met anybody who has diabetes, the way that diabetes works is if you eat sugar, right? Here's a graph. If you eat sugar, your blood sugar, the concentrated amount of sugar in your blood, rises. Normally, your body would begin to produce insulin then to combat the rise in sugar, and both levels would eventually fall off back down to normal. But with a person with diabetes, the blood sugar rises, but their insulin levels are not high enough to combat all the sugar. So if that sugar gets too high, then they can go into coma or have other issues. So insulin is used to manage blood sugar levels. We need to be able to mass produce that. We do make it, but not efficiently enough for us to use a human. Obviously, remember when a human cell divides, it's a much longer process. All right, now remember, DNA is a universal code for all living things. That should say things. Even if the bacteria doesn't have the same genes that we do, they still have the same nucleotide. So they have the same G for guanine, binds to C for cytosine. They still have adenine. They still have thymine. They still have uracil in their RNA. So remember, the same building blocks are in all organisms because DNA essentially is the same, even though their sequences to make different genes are different. Sorry about that, I'll say that again. Even though their sequences of these nucleotides might be different, the building blocks are the same. That's why DNA is universal. Why do you think scientists want to identify the genomes, map out the genes of other organisms? Think about that for a little bit. This table is showing us that of all of these species of organisms, bacteria, fungi, insects, plants, etc., this is the number of species we've discovered, right? So bacteria, there are 100,000 to 10 million different species. And within those, there's 17,000 different genomes. Whereas if you look at a vertebrate, like you are a vertebrate because you have a vertebra spinal column. So these are all organisms that don't have a spinal cord. Anyway, it's just showing you that even though there are the same DNA code, all these organisms are different. There's so much different potential for genes that we could use. So we want to discover the genome so that we can use them for genetic engineering. Now, genetic engineering is not the same as biotechnology in that it's changing the DNA of an organism. So this is where we get into the controversial area of genetically modified organisms. So it's when we identify a gene in one organism and we can transfer it to another organism. So these obviously these illustrations aren't actual food products, but you get the idea. Changing the colors of fish or berries or combining two fruits. There are a lot of pros and cons about genetic engineering, so pause the video and read through these yourself. But overall, the pros for genetic engineering is that we can reduce diseases. We can make vaccines. We can make preventative medications. Oh, medications, right? That would be number two. It's not just insulin, but hemophilia, blood clots, cancer. And then we can create new crops. And that's a pro because there's a lot of places on this planet that aren't um, fertile for growing crops. So maybe we can make crops grow better in poorer conditions. Some cons, depending on who you ask, genetically modified foods may have less nutritional value. There might be more allergies to them, and they're definitely changing, even over the last decade. Another con is that if you had put that potential in the wrong hands, you might have biological warfare. Okay, and then the third one is we can be reducing genetic diversity by engineering only the parts, only the adaptations that we prefer. So corn might not be as adept at fighting off pests and bugs as they used to be. All right, so one specific example we're going to talk about is something called BT corn. BT corn originally was genetically modified. There you go, GMO again, to resist insects. Digestive bacteria in termites are being studied because they convert plant material into ethanol as a waste product. So we can use that as fuel source for cars that isn't fossil fuels. So that's not a bad thing. That sounds like a really good technology that you guys, as your generation, can explore more for us. All right, so back to our learning targets. You need to be able to explain how DNA technology is used to construct recombinant DNA. So know those steps, right? Those you have to know for the test. And you have to understand how genetic engineering or biotechnology is used to produce novel biomedical and agricultural products. Specifically, think of the examples, BT corn and the ethanol bug back here. The ethanol and termites. I'm sorry, I couldn't have remembered my thought here. So remember, not all genetic modifications and engineering is bad. We just have to have ethical regs and rules and regulations. Okay. All right. Let me know if you have any questions. I will see you guys next time.